Hey, good morning. Um, so now that we've talked a lot about tumor suppressor genes and tumor suppressor proteins, I think it's a good time to go back and revisit some additional examples of oncoproteins. So as we build up our knowledge and our appreciation of how tumor suppressor genes and oncogenes are sort of in this constant back and forth kind of battle to determine what happens in our cells, with the outcome of hopefully maintaining control over cell cycle proliferation. And that's how our cells normally resist transformation. I think it's good to look at examples of both sides. Right. So we've already looked at certain examples of oncoproteins like SARC and RAS, and we mentioned MYC. So today, let's take a closer look at MYC and see why MYC is a bit different than SARC and RAS in terms of how it functions in the cell and why MYC is particularly dangerous as a proto-oncogene if things go wrong with its expression or its function. Let's start off by reviewing some of the key concepts that we've been discussing so that we can get back on the same page here. We've said that most fully differentiated cells have limited proliferative capacity, right? So your fully differentiated skin cells or the epithelial cells that make up the lining of your colon, once they've fully differentiated and become a skin cell or a gland cell, their interest in proliferating is usually, no longer present, right? Because they're more focused on staying differentiated, doing their job until they're just physically lost by your body or recycled. So we said that during transformation, events that lead to the de-differentiation, right, coupled to events that lead to increased proliferation, those are some of the hallmarks of cancer. We also learned that it's during G1 phase of the cell cycle, that's when the cells are actively listening to external signals that are telling them what to do. Right? The brand new cell that is the daughter cell following mitosis, it's kind of reset. Right? We said that after mitosis, it's kind of back to square one, and that cell needs to know, what is it supposed to be doing now? Right? Should I continue on in the cell cycle, or should I try to differentiate and use all the proteins that I know I'm supposed, or make the proteins that I know I'm supposed to make in order to be the particular type of cell that I am? That's all a function of what it's hearing, right? What it's receiving in the form of other types of signals arriving from surrounding cells that tell it what to do, especially, right, if we're talking about the cell being an epithelial cell dependent upon signals from the supporting stromal cells. So as a result, its activity during G1 can kind of go back and forth a little bit, depending upon how much mitogenic factors it's receiving at a given moment versus how many differentiation signals it's receiving. Right? But importantly, we said what? If it keeps receiving enough mitogenic signals that end up pushing it to create a lot of cyclin Ds, CDK4, and that starts triggering the production of another wave of proteins. Once it reaches a certain sort of critical mass of those proteins, if you want to think about it, then it plunges over the R point, the restriction point. And at this point, it's now going to go into S phase, and the cell is committed to seeing this process to the end, all the way through mitosis. We learned that RB, or the protein product of RB, is a gatekeeper, right? Tumor suppressor gene that guards this R point. And it does so by holding on or sequestering those E2F transcription factors that would otherwise be driving production of the next wave of proteins that would be needed to move into S phase, for example. We said that RB mutations or loss of heterozygosity or the presence of viral oncoproteins such as the E1A, E7, right? Those can interfere with the regulation of RB, right? And get in the way of RB properly controlling those E2F transcription factors. And if RB is not working, then those transcription factors go to work and the cell cycle keeps going around and around. We also said, importantly, that the phosphorylation status of RB, such as when it's hyperphosphorylated, right? When all of its 16 possible phosphorylation sites on it have phosphate groups, that takes RB out of the picture, right? And allows the cell cycle to move ahead unimpeded. So that brings us back then to this cartoon that we come back to time and time again. And today I thought we would discuss the function of MYC. Right, so here's MYC down here. It's an oncoprotein, or I should say more technically, it's a proto-oncogene, right? Because normally it's a very important part of our cell's function, but MYC pops up a lot as a strong oncogene in different cancers, right? Where something has happened to mess up the regulation of MYC. We had looked at things like RAS or SARC. Where's SARC? Okay, over here before. And so what I want to remind you of is look at the different sort of location that these different oncogenes are at in this cartoon. We said that SARC and RAS are more like supervisors, right? Whereas MYC is more like the factory worker. So we said, whereas before we thought RAS and SARC could be very powerful oncogenes because if they change their mind about what they're supposed to be doing, in other words, if there's a mutation that alters their shape, right, that could have impact on multiple different downstream pathways. But it turns out that MYC, as a lowly factory floor worker, right, is just as powerful, if not more, as an oncogene. So how would that be? That would be if this particular factory worker, right, isn't just in charge of working at this one station here, but maybe if his skill or his influence is felt on numerous different little assembly line stations throughout the factory, right? Therefore, if he's messed up, right, and starts doing something weird, right, if he's a transcription factor that's only used at, let's say, this particular assembly station, then only the product of this station would get messed up, right? But if in reality, his skill and his hands actually visit all of the different assembly line stations in the factory and he's messed up, then you can imagine his effect is just as powerful as his supervisor making a bad decision in the first place. Right? So that's the story of MYC. So let's take a look at that. MYC is a transcription factor. That's why in that cartoon, it's shown interacting directly with DNA. And remember, our sort of general description or definition of a transcription factor is some protein that either directly binds to DNA, right, or is very closely associated to those proteins that bind to DNA and function in a way to influence gene regulation. So these would be binding sites that are at maybe like promoters or enhancers, other types of regulatory sites that ultimately influence what's happening at the promoter. So certain transcription factors we can appreciate as being proto-oncogenic. So these are different classes of these genes that make these types of proto-oncoproteins. And they fall into different classes based upon their structure. You know, what it is in the structure of the protein that enables them to do something like bind to DNA. Some of them have structures that we call leucine zippers with basic DNA binding domains. Some of them have zinc fingers, in other words, peptide regions of the protein that associate with a zinc ion. And that zinc ion, that metal ion, helps hold the structure in a shape that can interact with DNA. Some of these are homeobox proteins. So many of the early developmental genes in the embryo have these homeobox type of domains in them. There are some miscellaneous ones as well. But here, the helix loop helix, or basic HLH domain, is a common type of protein structure that's used in proteins that belong to the MYC family. So let's look at what MYC does. So MYC binds directly to DNA as a partner in a dimer with another related protein. For example, in this illustration here, MYC is partnering up with MAX. So MAX, you can see down here, stands for MYC-associated factor X. Right? And MYC and MAX as a dimer recognize this particular DNA-based sequence that's often found in the promoters of genes. We call this particular sequence an E-box. All right, so the CAC-GTG This would be like the consensus. So throughout the genome, you'd find slight variations of this, right? But this is sort of like the most popular one would be CACGTG. About 80% of the genes in our genome have these E boxes present. Right? Wow. So that's why I was saying, right? Mick, as a factory floor worker, right? He's not in charge of just one particular station. About 80% of the genes in our genome have some sort of influence of Mick over them. Right? The binding of the Mick-Max heterodimer together on the promoter creates the right shape. So the two proteins together creates the right shape to recruit other proteins that are histone acetyltransferases, or HATs. Right? So HAT proteins that are floating by. Normally, if there's no Mick or Max bound to a promoter, maybe the protein would just float by, right? But if Mick and Max is there, then this histone acetyltransferase will bind to it. And what will it do? It'll start transferring acetyl groups to histone proteins that are nearby. Those histone proteins that form the nucleosomes that the DNA is wrapped around, right? So as a result, the chromatin in that region begins to unwind, right? And the more DNA that is unwound, or I should say the more chromatin that's unwound, the more available different DNA sequences are for other transcription factors to see the binding sites that they like, right? So that's how Mick and Max work. Ultimately, of course, that will recruit RNA Pol 2. So let's compare Mick with something like RAS to appreciate how sort of awesomely, simple Mick is, right? And yet how scary powerful Mick is at the same time because of its simplicity. So we looked at supervisory oncoproteins like RAS before. So this is not new. This is review. Hopefully it's review. Hopefully none of you are like, what's this? I've never seen this before, right? We said RAS hangs out on the underside of the cell membrane. Normally it's bound in its GDP bound state, right? So in that state, the RAS is the wrong shape to signal. But if you have different mitogenic factors like epidermal growth factors arriving and binding to receptor tyrosine kinases like an EGFR, then you'll get the phosphorylation of the tyrosines on the tails of the receptor that then start to coordinate the recruitment of different proteins that have things like SH domains that will recognize phosphorylated tyrosines, right? Or SH3 that recognize proline-rich regions. And that will eventually recruit things like these, like SOS, right? Guanosine exchange factor. And now this SOS, instead of floating around somewhere, it's brought to where RAS is, right? That's how this works. And SOS then exchanges the GDP to GTP. And when RAS is bound to GTP, now its shape is right to start signaling other downstream proteins. And then we said, well, then RAS can be turned off if you have other proteins like GAP that encourage RAS to do its GTP ACE activity. And it breaks the GTP and turns it back to GDP. And RAS is very well behaved here, right? It shuts itself off. Right? So that's pretty complicated. So as a result, RAS has to have all these different functional domains as part of its complicated 3D structure in order for all of these messages and signals to actually happen. I mean, I think that is really amazing, right? That's when I always say that these proteins can be complicated machines, right? That have on and off switches and input output types of domains. So when you look at RAS here, so this is the ribbon structure of RAS, and it's showing its guanosine nucleotide binding region. So this is GTP, right? So there's one phosphate group, two phosphate groups, three phosphate groups. If you were to do a space filling, excuse me, space filling model of this, here is RAS. Now it's actually shown bound to GDP, right? Because now there's only two phosphate groups there. So the shape of this is a bit different than the one on the left. And if you overlapped different RAS family members and color-coded them, what you would find is that certain regions of RAS proteins are the most highly conserved in terms of amino acid sequence in the nucleotide binding clef over here, right? Because they're all GTP aces, but variations in amino acid sequences elsewhere in the RAS family proteins, like NRAS, HRAS, KRAS, right? The differences in shape over here are what determine different downstream protein partners that each one would communicate with preferentially. Right. So that's super cool, right? So you look at the structure and hopefully you're like amazed at the thought, right? That this is coded for by our genes for RAS, right? And that it's the sequence of amino acids that create all these complicated on-off switch regions and input-output domains of the protein. So if this is RAS, what does MYC look like, right? If MYC in many ways is just as powerful and can talk to 80% of our genes. You might think, wow, MYC must be even more complicated, right? Have multiple different input-output pathways in order to function, on and off switches, things like that. The reality is, and this is what makes MYC so cool and so scary, is that it's the exact opposite, right? This is the structure of MYC, right? And in fact, this is not even MYC alone. This is MYC partnered with MAX. Right? So MYC in red is this part that comes down here from the top to bottom, goes around, and then continues here, and that's the whole protein, right? But you can see this is why it's called a basic helix loop helix, right? And that's it, right? And you're like, that's crazy, right? How could a protein that's so simple, right, have such tremendous, profound effects on gene regulation? So here it is partnered with MAX, and you can see the way they partner together, they are like a pair of twist ties twisted around the DNA. Each one of them have part of their helical structure that is perfectly nestled in the major groove of DNA, right? If the underlying DNA sequence is that E-box, CACGTG, then they will fit very nicely across the bases down there. Right? So here it is as a space filling model. In fact, here are two dimers, right? Each one of these is like a twist tie of two partners together. So here, for example, let's pretend that the red one is MYC. So it's a helix that comes down, a loop, and then the other helix goes behind the DNA molecule here, right? And the other one is MAX. Here's a helix, there's a loop, and then a helix. Just like a twist tie twisted around the DNA. Right? So you say, well, how does that work? Well, this three-dimensional shape on the top that forms is the right shape so that a histone acetyltransferase protein that's floating by will bind to this and stop and hang out on the DNA at this spot, right? And therefore start acetylating histones that are also in this region, okay? So I think it's remarkable, but that's why I say, right, it's both awesome in its simplicity and also terrifying in its simplicity because there are no on and off switches here, right? There is no on and off switch. MYC just functions as long as it's being expressed. Okay? So the balance then between how the cell is controlling things like proliferation going through the cell cycle versus hanging out in G1 and focusing on differentiation has a lot to do with how MYC is behaving with its partner proteins. So we said when MYC is partnered up with MAX and they are bound to their E-boxes in the promoters of certain genes, right? Then that will favor the acetylation of histones in the chromatin in this area, and this promoter becomes more exposed, and other transcription factors then start seeing their binding sequences, and they start piling up, and RNA Pol2 ends up being recruited, right? So many mitogenic genes, right, that are responding to mitogenic signals that are coming from the outside of the cell are controlled by MYC and MAX in this way. For example, cyclin D, right? The cyclin D gene has MYC and MAC E-boxes in its promoter, right? So as you get more mitogenic signals, right, MYC and MAX then go to these genes and they begin to create things like more cyclin D, right? That will do things like encourage G1 to start moving towards S phase. On the other hand, right, if the signals that are arriving on the cell are more inhibitory in terms of proliferation, right? In other words, signals that encourage more differentiation, So it wants the cell not to bother making more of these proliferative cell cycle genes, but it wants the cell to make more of the genes that are involved in differentiation, right? So that the nerve cell knows I'm supposed to make proteins that help me be a nerve cell, rather than, oh, I should just be like rushing through the cell cycle, right? So that's why differentiation and proliferation are like a seesaw, right? The cell is either doing one or focused on the other. So if pro-differentiation signals arrive, the cell produces a lot more of this other protein, mixed. And mixed then will preferentially partner with max, right? And different genes on their e-boxes will get the combination of mixed and max, right, instead of mic, right? And as a result, this then will help to shift the balance of which genes get expressed. So this mixed is kind of a funny story because originally, for many years, this protein was not called mixed. It was actually called MAD, right? And so it was all about Mad Max, okay? And so the story goes, there's no particular reason why it was named MAD, other than the people who discovered it thought it would be hilarious because it partners with Max. So you'd always be talking about Mad Max, right? By the way, that movie, one of my favorite movies of all time, awesome movie, right? Which is actually good because it's like totally like this driven, aggressive movie. When you think about like oncogenes, it kind of fits. All right, whatever. So later they changed the name of MAD to mixed. Okay, and there's actually a good reason they changed the name. And it wasn't because they were tired of making this movie reference. Let's look at what's going on here. Let's go back to our example of the colonic crypt, right? So these are the little crypts that are in the epithelium of your colon, and these crypts are where stem cells at the bottom are encouraged to proliferate so that they're always replenishing the number of fully differentiated epithelial cells that line the surface of your colon. And these fully differentiated epithelial cells, they produce mucus, they have cilia to help absorb things like water, etc. Right? So we looked at this cartoon on the left before, and we studied it in the context of understanding how beta-catenin signaling is influencing this shift from proliferation to differentiation. Remember, we said at the bottom of the crypt are these stem cells that are receiving Wnt signals, the little red arrows, from the supporting stromal cells that are beneath the basement membrane, right? And just as long as they're receiving a lot of Wnt signals, remember Wnt, which is received by the frizzled receptor, will discourage the complex that degrades beta-catenin. Right? So as a result, beta-catenin is allowed to signal and function and turn on genes that control proliferation. Right? So as those cells proliferate, they're physically pushed up and away from the basement membrane. So they get less and less of the Wnt signaling, and then you get a shift. Right? You get a shift away from proliferation, more towards differentiation genes start turning on until eventually, right, you have the fully differentiated green cell at the top here, and it does its job for a few days before it's lost. Right? So let's look at expression levels of what's going on inside here. So during the proliferation activity down at the bottom, you have a lot of MYC working with MAX together on the promoters of proliferation genes, right? So these are genes that are stimulating cell cycle progression and division of the cell. And that is largely a target of the beta-catenin. So the beta-catenin will help influence MYC production, right? So as MYC production is being made, then you'll have more of this MYC and MAX coming together as twist ties on certain genes, and then those genes will preferentially be activated because at those particular genes you will have more acetylation of histones, right, and more RNA Pol2 activity. As the beta-catenin signaling goes down, right, as the cells are pushed farther and farther away from the Wnt signals, then you get a shift. Less MYC is made, right, and more of the mixed is made, the green thing, right? So now mixed will partner with MAX, and then the influence it has on different genes is it will favor now the expression of genes that are more having to do with differentiation. So in other words, the genes that are meant to be the genes that code for protein products of cells that are colonic epithelial cells, right? So that's why this dotted line here, it's kind of neat to think about. This staining image over here is cool because what they're doing is they're staining for two different protein markers that are associated with, for example, in green, they are staining with an antibody that looks for Ki67. So Ki67 is a nuclear protein that's expressed by cells that are undergoing proliferation, undergoing mitosis. Right? And so as a result, the cells that are in the lower part of the crypt, right? So just in case you have trouble telling what this is, the dotted lines represent different sort of parts of the crypt as you come down to the bottom, right? Different sections. And so these cells that are towards the bottom are more concerned with proliferating, but once they get far enough away from the Wnt beta-catenin signaling, right, and once MYC starts being expressed less and less in them, right, then they shift over to differentiation. So this red stain is this Krebs protein that's a marker for differentiation. Right. So what does that imply? That implies that the cell is always in this kind of back and forth seesaw between proliferation decisions versus differentiation decisions, right? And that even though this is not a black and white on-off type of situation, right, it is certainly representative of major shifts in the waves of different proteins that are being made, right, that will determine what the cell is actually doing at that given time in G1. One way I think about this is if we want to talk about oncogenes as being sort of like accelerating signals, right, driving the cell cycle forward, and tumor suppressor genes as being the brakes. We already discussed this analogy that in your car, right, it's way more important that your brakes work, that you have 100% braking capability rather than always worrying about having 100% acceleration capability, right? Because you can still drive your car even if the acceleration is kind of lousy, but you cannot safely drive your car if the brakes have failed. Right? That's why, again, we evolved many more different tumor suppressor pathways than necessarily clever different oncogenic pathways. But the car is like a car being driven by a two-footed driver then. Right? Some people are like this. I don't know. I usually drive my car with just my right foot, and it either goes between the brake or the accelerator, brake or the accelerator. Some people, I think they're very sort of anxious, and they actually have one foot on each, and they're actually constantly kind of pressing both. Right? I know that sounds kind of, it's not very safe, and it's not good for your car. Right. But a lot of times you'll see cars on the highway that always seem to have their brake lights on. Right. And these are these sort of anxious two-footed drivers. They're always applying some brake, always some gas. It's whichever one they happen to be pushing harder at a given time that determines what their car is doing. Right. So our cells are like this as well. Right. Because hopefully one thing you've come to appreciate is you have these proto-oncogenic proteins that are doing things to try to drive the cell forward. At the same time, you have these tumor suppressor proteins, right, that are trying to hold it back. So let's see how something like MYC plays a role in this. So here on the left, we've talked about this before, right? Let's start from the bottom here, right? Let's say in order to get a cell pushed into S phase, the cell has to get pushed beyond the restriction point in late G1. So let's work backwards. What's pushing the cell beyond this restriction point? It is the activity of that family of transcription factors, the E2Fs, right? That will be busy causing the expression of different genes and protein products that are the next wave that's going to push the cell over the restriction point. How are those E2Fs normally kept shut down? Through the actions of RB. Right? So we have accelerators like E2F, that's the gas pedal. Right? We have brakes like the RB, right? Very important gatekeeping tumor suppressor gene. Right? But then you have other things working on RB to try to eliminate RB, right? So while you have RB trying to inhibit the E2Fs, you have things like the cyclin and CDKs beginning to phosphorylate RB more and more, right? And the more phosphorylated RB is, the more inhibited RB becomes. So we said that ultimately high levels of cyclin E and CDK2, that's going to be the real kicker that causes RB to be fully hyperphosphorylated in the cell so that RB lets go of the E2Fs, right? And then the gas pedal is pushed down and the car moves forward. So RB is also acted upon by cyclin D and CDK4, right? Those are the first sort of cyclins and cyclin-dependent kinases at the start of G1 that begin building up and start encouraging RB, at first hypophosphorylating it so that it works, but then gradually hyperphosphorylating it as well. Right? So we said earlier in this lecture that cyclin D and CDK4 is a prime target of the MYC-MAX combination, right? So MYC and MAX activity is going to drive cyclin D forward. So as RB puts the brakes on E2Fs 1, 2, and 3, MYC and MAX is trying to accelerate by pushing more cyclin D. And MYC and MAX also creates the expression of more E2F proteins, right? So the E2F proteins, their genes have these MYC-MAX E boxes in them, right? So how does the cell, right, fight back? How does the two-footed driver then increase the brakes? So the two-footed driver, your cell has stuff like P15, P21, P27, other tumor suppressor genes, right? They go to work by trying to inhibit the activity of the cyclin CDKs. Right? So another layer of braking on top of a layer of accelerating. Right? But then MYC, with continued MYC expression, MYC pushes things even harder. So MYC and MAX also results in the expression of MIS1. Right? And then MYC and MIS1 combinations will bind to the promoters of P15 gene, P21, and P27 gene and prevent them from being expressed. Right? So hopefully what you see here is it really is like a two-footed driver. Right? Every time there's a layer of accelerating, there's another layer of braking that your cell has to keep that in check. But above that layer of braking is another layer of accelerators to undo the braking. Right? And then above that is another layer of brakes to keep those in check. Right? So in conclusion here, MYC, because it's just this lowly little transcription factor that has widespread felt influences on multiple genes, MYC is a critical player in these multiple layers of braking and accelerating. And as I said, the scary thing is because MYC itself, the protein, does not really have an on and off switch, right? It just has to do with how much MYC you have in the cell. This makes MYC very powerful, right? And now you begin to realize that's why MYC shows up as one of the most common oncogenic mutations in so many cancers, right? Because if you lose control over MYC functioning like this, you're losing control of major groups of genes in your cells. All right. So just as an example, so here we have a section of Drosophila chromosome 3. So this is a fly chromosome. Right? And what they've done is this section of this fly chromosome, maybe there are certain genes in it. I believe maybe these individual gray lines might be individual genes, and many of these genes might be involved in things like tissue growth, proliferation. I think in this particular case, the illustration is they're going to show you that has to do with vascularization, right? Genes that actually determine whether blood vessels will grow to feed and supply tissue with nutrients and oxygen. Okay. So what they've done here is they've actually mapped out along this section of chromosome 3, can you see where there are different spots where MYC in green or mixed in red, in flies it's called MNT, but it's the same thing as mixed, or MAX in blue. And you can see, sure enough, you see different examples where MYC will partner up, right, with either MINT or MAX and as a result, have different influences, right? Depending on who's partnering up with who, that will have an influence on what happens at this gene. So how important is MYC? Look at this example, right? So this is a tumor that arose from mutated mouse stem cells. This is a tumor that was then isolated from the tissue. On the left is a tumor that was raised in a wild type mouse that is wild type for both copies of the c-MYC gene, right? From the copy that it got from the mom mouse and the copy that it got from the dad mouse, right? And this is the extent of vascularization of this tumor. And as a result, the size of the tumor, because it has a healthy supply of blood, providing nutrients and gases. In the same type of mouse strain, here they've knocked out the MYC gene, right? So this particular mouse is now devoid of the MYC gene, right? So it's double negative knockout. Right? And as a result, when a stem cell tumor is encouraged to grow in it, if both mice are given the same amount of time to develop the tumor under the same conditions, look at the dramatic difference. Right? So without MYC signaling available, right, this is the impact on tumor growth and vascularization. So MYC is also controlled to some extent by other proteins. Now, I had said that the scary thing about MYC is that it doesn't really have an on and off switch like something like SARC does or something like RAS does. Right? But certainly there are other protein pathways that sort of intersect on MYC and the production of MYC. So in Drosophila, there's a gene known as DPP. It's actually called decapentaplegic. Right? So decapenta means 15. Right? So it's named this way because the normal functioning of this protein leads to the normal formation in the early Drosophila embryo of the 15 different tissue segments that become the body, the legs, and the wings. If there's a defect in this DPP gene, right, then this is the type of fly that arises. So very clearly, morphological defects that can be traced back to the early development. There's another protein made by a gene that's called Mothers Against DPP, right, or MAD, right? So you can tell people are getting a little too clever for their own good here, that affect DPP. So it was identified earlier that this MAD protein is a regulator of the DPP, and then DPP itself has some influence on control over MYC. Okay. So I wanted to point out that because this was named MAD in flies, right, and this is a very different protein than the MAD that was originally identified as the MAD-MAX, MYC partner, right? You could tell how this would get confusing really fast, right? Because both of these are somehow involved in the regulation of MYC. But this MAD protein, Mothers Against DPP, is not the same one that binds to MYC. So that was not the same MAD. So that's why human geneticists decided, all right, joke's over. We can't keep calling this MAD. We better change the name to something else, right? So they changed that protein to mixed, MXD, so that they could still call this one MAD. So in humans, right, we have SMAD, right? So it's a homologue of the Drosophila MAD. And because we have this SMAD, we want to change the name of the original MAD protein that partners with MYC to mixed instead. So how do these SMAD proteins work? Right? They work in response to signals arriving from the outside of the cell that discourage the cell to proliferate and instead encourage the cell to differentiate. Remember, it's that seesaw again, right? Signals that favor differentiation do so at the expense of proliferation and vice versa. So when transforming growth factor beta arrives at TGF beta receptors on the outside, you can see what's going on inside the nucleus here. So you have in the nucleus here, you have MYC, right? MYC that's partnering with Ms, right, and wanting to influence this gene here. Part of what this gene's influence does then, it is also inhibits things like P21, right, which is inhibiting CDKs. Over here, you have P15s that were trying to inhibit cyclin D CDKs, right? So if MYC and MYZ are allowed to function over here, right, you are driving the cell cycle forward by discouraging the functions of this layer of tumor suppressor genes. So when TGF beta signals, TGF beta signals, and it signals through these SMAD proteins. So SMAD2 and 3 and SMAD4, they come together and they end up partnering with MYZ1 instead of allowing MYC to do so. Right? So here you can see they are then interfering with MYC, not because they're somehow messing with an on and off signal of the MYC protein itself. They are just preferentially binding as the partner to MYZ1 and booting MYC off. Right? And as a result, that alteration in gene expression leads to the increased production of tumor suppressor genes like P15, while at the same time taking away these inhibitory signals that are coming from the original MYC and MYZ. Right? So SMAD proteins also form a repressor complex. So the SMAD protein here will associate with the RB family protein P107, right, to really help sequester E2Fs, right, and prevent the expression of MYC. So here we're seeing the MYC gene, right, and the MYC gene is responsive to E2Fs normally. So if you have the production of SMAD proteins, that helps encourage the RB family proteins to more successfully sequester or keep the E2F shut down. So that, of course, will reduce the amount of MYC mRNA that's being translated into MYC proteins. Right? Okay, so the last slide for today then is this one. And I know you're like, oh, the last slide, why does it have to be like the busiest one? I don't want you to be like all overwhelmed by the slide, right? Because I don't expect you to memorize this slide, but I think it's a really cool slide because it helps us visually put some things together here. We've been talking about multiple different pathways of oncogenes so far, multiple different pathways of tumor suppressor genes, but I said, think of them as a two-footed driver, right? It's not either or, right? It's both sets of pathways, pro-proliferative pathways, pro-differentiation pathways that are all happening at the same time. And it just so happens to be whichever one, right, is more strongly pushed by the signals that are arriving on the outside of the cell that will ultimately determine what the cell phenotype is, right, what the cell is going to do next. So let's just have some fun looking at this and recognizing certain things that we've talked about before. Oftentimes with these types of pathways that have multiple like arrows and multiple sort of inhibitory ends like the T's, right? It's useful not to start at the top and get all confused, but to actually start at the bottom, right? Because you ask yourself, what ultimately is the determining factor on whether the cell moves forward out of G1 into S phase? And that is whether the cell, right, builds up enough waves of different proliferative proteins to push it over the R point, right, in late G1. So let's work backwards from that. So what's preventing the cell from crossing the R point? Oh, it's our friend RB, right? This very important gatekeeper tumor suppressor gene, right? So if RB is not allowed to do its job, then the cell will move forward. So how is RB not allowed to do its job? Well, if it's interacting with things like viral oncoproteins like E7, right, that bind to RB and prevent RB from physically interacting with the E2F transcription factors, but other things as well. For example, cyclin E CDK2s are inhibiting RB because they are hyperphosphorylating the RB. Right? And then you can move backwards. E CDK2, it itself is trying to be held from working, right, by tumor suppressors like P27 and P21. Okay. P21 and P27 themselves can be turned off from working if you have oncogene pathways like AKT inhibiting them from working. Right? And then what makes the AKT phosphokinase B pathway work? Well, it's receptor tyrosine kinases, right, that are responding to mitogenic factors coming in from the outside. Let's go to the left here, starting again from RB. We have things like MYC that are working, right? MYC that are working to do things like increase certain inhibitors of RB. MYC is also working to increase these factors over here that are inhibiting tumor suppressor genes like P27. MYC is also working to inhibit P15, right? So MYC, again, is the lowly factory worker who unfortunately has his hands in many different pathways. Right? And on the left here, right, probably the most sort of straightforward thing to remember is that the function of RB is largely dependent upon the activity of cyclin D and CDK4 and 6 from the start of G1 all the way to the end of G1, right? The cyclin D CDK6 activity is what is not only influencing RB through phosphorylation, but also interacting with many other gene pathways that are controlling RB. And where does that come from? It comes from the expression of the cyclin proteins, right, that are being influenced by things like Wnt signaling that inhibits APC so that you cannot form the proper complex that would destroy beta-catenin. So if beta-catenin is not destroyed, it goes to work and it turns on the production of lots of cyclin D, mitogens that are going through receptor tyrosine kinase pathways, activating RAS and through RAF, right? Also mitogens activating things like AKT, PKB again, over on this side of the figure, right? And that itself has another layer of brakes and accelerators above it, right? Here is PI3 kinase driving AKT, PKB as the accelerator pedal. Here is P10, right? Our friend P10 trying to stop AKT from happening by reversing PIP3 back to PIP2. And again, look, we have MYC over here also influencing the expression of cyclins, right? So I think this is a cool picture, right? Not because, oh, I need to memorize this or something, but I get it when I see this constant interplay between the blue pathways, right? The blue players in this pathway that are the tumor suppressor genes or the pro-differentiation signals, and the pink, or yeah, I guess pink oncogenic pathways, right? And they're all tangled up in this complicated pathway of many intersecting layers, right? So that's why I think it's beautiful, because it shows you, right, how complex cell signaling can be. And it really highlights this concept that the cell is constantly in this sort of flux, right? Depending upon what signals are coming from the outside, that will lean more heavily the orange peach pathways here or the blue tumor suppressor pathways, right? And that ultimately is going to determine whether the car moves forward or it stops, or in this case, whether the cell cycle moves forward or the cell is kept in G1, right? But I want this to also highlight to you how critical the RB is here, right? Because look at this, no matter which pathways you're following here, ultimately it all converges on RB, right? And that's why RB is such an important tumor suppressor gene. All right, let's go ahead and stop there.